ले ला दमन ले आ जाके मेरी क्लास चल रही है मैं ले आ देना पानी तेन चौंकड़ी मार चौंकड़ी मार चार्ज लाना है मेरी तो भी क्लास है दूध जा हाँ जी हाँ जी सुन दिया तुमने सुन दिया सिरोटी खाओ अपने इत्थे ही है तुमने इत्थे ही है हाँ जी दमन तुमने क्लास लेना चुप कर जो साइलेंट थैंक यू hello there hi everyone thank you for coming at this time also I hope everyone in the class, they are joining the classes later also. I can see the views are increasing, mainly the people who are part of this Telegram group. They're attending these classes. So I'm happy that everything is going satisfactory and all is well so far, with a good pace and good knowledge. And yeah, I hope you liked the previous session that we had about protein isolation and purification. I really prefer this part because this is a highly recommended part. The reason behind it, that after your DNA and RNA, your protein is being made. So all the research, if you do at the protein level, your level of, you know, the cost of your research will be more in the world. Of course, ribosomal RNA, RNA, microRNA, siRNAs, they also had very big importance. But most of the big labs in the world, they are working at the protein level at the end. Yeah, so that's why I give this part very preference. Thank you, Sojit. Thank you, Somi. We're trying our best that we give you the best so that you can also give the best to others. Yeah, so it's like I'm giving you the knowledge, so you have the knowledge, and somebody asks you to perform these assignments, these tasks of molecular biology. You need to perform even the best towards them. The person will say that okay, he or she is the person, I asked him or her to do this part and they did it very well. Yeah, so it's feel very satisfactory and feel proud, and yeah, that's the life. So on that note, it's already 21:05. I think I will write one message one time more and then we can start. Okay, this protein isolation and purification has been shared completely, so you can have a look about this presentation. All the videos are included in that. If your videos are not playing, then let me know. I will share the videos also later. Cool. So after this part, I will play one gel electrophoresis, one more different types of gel electrophoresis. Then we will discuss the various applications of all this technique from the industrial perspective. So be with me, and then we start. So that's not, yeah. Now next technique, which is very important in the protein separation, is your Marley-Toff, yeah, which is a matrix assisted laser desorption ionization, right? So laser beam ionizes protein embedded in matrix. Ionized protein accelerated towards a detector, and detector monitors the arrival of ions as the laser triggers the clock, and then TF is a time of flight. So what happened? You first you put a protein sample over the matrix, yeah. Then you put laser beam on top, and after putting the laser beam on top, you put the laser via this prism. There's a beam which trigger this laser, which gives which like get intensed over the your protein. Then due to which the ions are released in the positive form through the flight tube, and they get shorter and shorter as they fly over the other place because of the magnetic field being applied there. So this is electric field accelerated ions here. Then they get detected, and then we read the results about it. So this way we get peaks. There's an intensity and there's a mass is to charge ratio, right? So the intensity is 1 plus H, this is insulin, 5733.9, and there is a beta lactoglobulin, L plus H, 181364. Yeah. So in this way we can separate our molecules and we can find these peptides that within the sample these peptides were present. Some results of that. So let's see how it is done.
Brooker Multi-Toff process. Multi-Toff analysis provides additional structural information for target molecules. As multi-Toff is a two-phase process. Fragmentation can occur by application of higher laser energies, known as LID, laser-induced dissociation, or a background gas can be introduced to promote ion collisions. This process is known as collision-induced dissociation, or CID. Samples are co-crystallized with a suitable matrix on a sample carrier. The samples are vaporized by the laser into a vacuum while being ionized at the same time. During this process, the sample molecules can become excited. The now charged and excited particles are accelerated through the application of a high voltage, and fragmentation can occur as they travel through the flight tube. In this process, precursor ions and their respective fragments travel together at the same speed. Based on the expected time of flight of the precursor ions, specific ion packages may be individually selected, and all other ions blocked from continuing through the flight tube. The selected ions are then re-accelerated, with the precursor and fragment ions traveling at different speeds towards the reflector detector. The ions travel via the reflector towards the detector, and thus a top-top spectrum is recorded. The detection of the selected precursor ion and its fragments creates a top-top spectrum. Whether fragmentation occurs through the use of higher laser energies or a collision gas, the structural details derived from the multi-top spectrum provide a deeper analytical insight of the targeted compound.
Okay. Yeah, this video I will play later. So now we will talk about electrophoresis. So some protocols may be beneficial for you. All good. So maybe in future you might be doing some research in your hemisphere. Uh, you can use these protocols for your research. I have actually many protocols. Where are they? Okay, maybe later when I find them I will share with you. So now as we have just touched a little bit about of electroforces, now I will go more detail about various types of electroforces that are present, which is being taught in the field of uh, okay, connection is a bit poor. Anyhow, let's see till when we can continue, otherwise we go to Google Meet. um, so yeah, more about electroforces now. Okay, So electrophoresis technique. So basically electrophoresis is a physical method of analysis which involves separation of compounds that are capable of acquiring electric charge in the conducting electrodes. Yeah. So electrophoresis may be defined as the migration of the charged particles through a solution under the influence of external electric field. So ions that are suspended between two electrodes tend to travel towards the electrodes that bears opposite charges. So there's two types of electrophoresis that we're going to discuss in this lecture. That is zone electrophoresis and moving boundary electrophoresis. Yeah. In the zone electrophoresis, it has paper electrophoresis, gel electrophoresis, thin layer electrophoresis, and cellulose acetate electrophoresis. And in the moving boundary electrophoresis, it has capillary electrophoresis, isotopes, isoelectric focusing, and immunoelectrophoresis. Yeah. So in total, 8 types of electrophoresis are present. So these things are present in your subjective book, in your book, but in practically, you mainly use your agarose gel electrophoresis, and you use your 2D gel electrophoresis, right? And you make your SDS page. One of that thing is done, and capillary electrophoresis is also being used. So it's really depend upon how much work you want to do and in which field you want to do. You can use that type of electrophoresis. Okay. Then comes your zone electrophoresis. First thing first, it involves the migration of charged particles on the supporting media. So paper, cellulose, acetate membrane, starch, gel, polychromolite. So all these come under this category, and it has a component separated but distributed into discrete zone on the supporting media, and supporting media is saturated with the buffer solution, and small volume of sample is applied as a narrow band. On applications of potential difference at the end of the strip, components migrates at a rate determined by the electrophoretic mobility. They are useful in biochemical investigations. Small quantity of sample can be analyzed, and cost is quite low and easy to maintain. So in general, what happened, you have a saturation of medium with the buffer, sample application, then electrophoretic separation, then removal of the supporting media. So then instrumentation that we required in this is your electrophoretic chamber, electrodes, diffusion barriers, supporting and stabilizing media. So in paper electrophoresis, you are having this 45 volt in DC. You have your electrolytes, you have your two glasses in it, two glasses plate. Yeah. So you have a pencil line, you put your sample in that, it's touching the both positive and negative end, and an apparatus for the electrophoresis. So horizontal paper electrophoresis and vertical paper electrophoresis are being used within the paper electrophoresis. The horizontal one apparatus looks like this. There is an airtight housing on the top, and your filter strips are added onto this, and you have a buffer solution having the electrodes positive and negative. Then comes your vertical paper electrophoresis, and it has a triangle shape like the structure with a sample application point on the top with an airtight housing with electrode positive and negative at the end. So filter paper such as Wattman number 1, number 3 millimeter in strip of 3 or 5 centimeter wide has been used to good effect, and separation take around 12 to 14 hours. Then continuous electrophoresis. So sample reservoir. So you have a buffer solution and small components which are having electrodes and filter paper. So you get your samples. This is used for preparative scale purpose. Predetermined sample volumes of 0.2 milliliter per minute through the wall device is applied. Silica gel, powered glass, sand, and agar gels can be used. A potential difference of 500 volt is applied, and pure compounds are collected in separate containers. The solvent is evaporated and pure fractions are reused afterwards. Then comes your gel electrophoresis, in which separation is brought about through the molecular sieving technique based on the molecular size of the substances. So gel material acts such as molecular sieve here, and gel is solid, is colloid in solid form, that is 99% is water, and it is that we have just discussed gel electrophoresis in our previous lecture. Same way, you put your gel, you put your comb, and then you put your sample and give your power supply, and your samples get separated according to the size. And this gel electrophoresis could also be two type, vertical and horizontal, and we can use two different gels, agar and agrose. So main difference between agar and agrose gel is that agar is a mixture of polysaccharides extracted from the seaweeds, whereas agrose is a highly purified uncharged polysaccharides derived from the agar itself. It's a byproduct of agar. Yeah, and agrose is a chemical based disaccharide repeating units of 3,6 anhydro L-galactose, and agrose dissolve when adding to boiling liquid. It remains in liquid state until the temperature is lowered to 40 degree, at which point it gels. So agrose gels are fragile, they are actually hydrocolloids, and they are held together by formation of weak hydrogen and hydrophobic bonds. And advantages are they are easy to prepare, sample concentration of agar is required, the resolution is superior to that of filter paper, and large quantities of proteins can be separated and recovered. So some disadvantages, electro-osmosis is high in them, resolution is less compared to polychromolite gels, and different sources and batches of agar tend to give different results, and purification is often necessary. So application is widely used in immunoelectrophoresis. So what happened to the agrose structure? You have a solid state, then you give 45 degrees Celsius, they started to make this dimers. Then with the aging, they give a final gel structure. Then you have your initial gel, then you have a solid state afterwards. So let's see how the agrose gel electrophoresis is done practically now.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer. Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button and observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the sample spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on, then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. At this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
Yeah. So this was all about agarose gel electrophoresis. Now comes your polychromolite gel electrophoresis, that is your page. It is, as we have discussed in previous lecture also, it is a polymerizing acrylamide monomers in presence of methylene bisaccharomelite to cross-link the monomers. So structure of your acrylamide CH2, CHCO, NH2 with N, and polychromolite gel electrophoresis is held together by the covalent cross-links. And polychromolite gels are tougher than the agarose gels, and it is thermostable, transparent, strong, and relatively chemical inert. And gels are uncharged and prepared in variety of pore sizes. And proteins are separated on the basis of charge to mass ratio and molecular size phenomena called molecular sieving. So some advantages. Then types of page, it could be native page or denatured page or SDS page. So the native page, they run under the non-denaturing conditions that does not have any SDS or DDT, and the separation is based upon the charge, size and shape. And this was the original mode of electrophoresis initially, but recently, to separate your molecules upon the molecular weight, so the common method was to determine the molecular weight of proteins. So in that case, your denatured or SDS page are used. So page we have discussed already. You run your gel under the page. You see the results with the ethidium bromide. You can see your result under the ultraviolet ray. And then within this page you add SDS, sodium dodecyl sulfate, which covers over your proteins, give a negative charge and helps to protein migrate according to their increasing molecular weights. So it just give a negative charge to your proteins, make it linear. You add your reductant SDS and heat it to your native protein, and it's good to go then. Then starch could be also used as a granular starch, should be boiled in a buffer to give clear colloidal suspensions. The suspensions on cooling sets are semi-solid gel due to inert weaning of the branch chains of amylopectin. So in order to avoid swelling, shrinking, petroleum gel is used. Then comes your vertical and horizontal gel electrophoresis. These are the methods for applications of sample. Sample is mixed with the gel by soaking a bit of filter paper in sample and pressing it into the gel. And detection by staining or by direct gravitimetry or UV absorption. Then comes your pulse filled gel electrophoresis. So pulse filled gel electrophoresis is a technique which is used for separation of large DNA molecules, which are very big ones, around 30 to 50 kilo base pairs. And what we do is, in this we periodically change the field of direction of your electric field so that various lengths of DNA react to change at different rates. And the voltage is also periodically switched among three directions. So one that runs through the central axis of the gel and two that run over the 60-degree angle sides. So like this. So it will give electric field on this side, on this side, on this side, and then your protein, your DNA will be separated according to that. Then comes your thin layer electrophoresis. Studies can be carried out in thin layer of silica, Kyle sugar, or alumina. It's take less time consuming and good resolution, and widely used in the combined electrophoretic chromatography studies in two-dimensional study of proteins and nucleic acid hydroxylides. Then comes cellulose acetate electrophoresis. It contains 2 to 3 acetyl groups per glucose unit, and its absorption capacity is less than that of paper. And it gives sharper bands, provides a good background for staining your glycoproteins. And further, moving boundary electrophoresis. The moving boundary method allows a charged species to migrate in a free moving solution without the supporting media. The instrumentations that we require is consist of a U-shaped glass cell of rectangular cross sections with electrodes placed on one of the limbs of the cells. Then sample solution is introduced at the bottom or through the side arm, and the apparatus is placed in constant temperature bath at 40 degrees Celsius. And detection is done by measuring refractive index throughout the solution. So here we have anode and cathode in this U-shaped chamber. You have your initial boundaries, you put your samples, and as the migration starts, they start to migrate according to their charges. Then comes your capillary electrophoresis. In the capillary electrophoresis, this is the highly technique used for the separation of your molecules actually based on the physical characteristics. So in the capillary electrophoresis is a technique which is performing electrophoresis in buffer filled narrow bore capillaries. So normally from 25 to 100 millimeter in internal diameter, and high voltage is applied in them, and capillaries are typically 50 micrometer in a diameter and 0.5 to 1 meter in the length. So here we can see we have a capillary electrophoresis going on with the cathode and anode. We have positive and minus molecules here with the buffer end on the other side. Yeah. So what you do is you have your absorption with 280 micrometer, and time is written here. Yeah. So they start to separate according to their thing, and then we can get the results. So high voltage injection, the potential is applied across the sample to enter the capillary by combination of ionic attraction and electroosmotic flow. In the pressure injection, pressure difference is used to drive the sample into capillary by applying vacuum into it. Then electroosmotic flow, the surface of the silicate glass capillary contains negatively charged functional groups that attract positively charged counter ions. So the positively charged ions migrate towards the negative electrode and carry solvent molecules in the same direction. And this overall solvent movement is called electroosmotic flow. So you have your negative charge on the top and you have a positive being attached to them. So your positive and negative charges will flow in between and they will give a electroosmotic flow. Yeah. And small volume of sample is moved into one end of the capillary, and the capillary passes. So here we have capillary tube. In its center we can detect the data in presence of your buffer electrolytes. Then comes your isotopes. Isotopes, it's a technique development based on the development of your potential gradient. So in this, it's also based on the moving boundary electrophoresis. A leading electrolyte, for example, chloride with higher mobility than analytes, and a trailing electrolyte with a low mobility are used. So solutions in which separations takes place is normally an aqueous medium which contains sucrose to provide higher density to the solution, where the separation by isoelectric focusing depends on the existence of pH gradient in the system. So the technique of isotopes depends on the development of a potential gradient, and separation of ionic components of the sample is achieved through the stacking them into the discrete zones in order to mobilize, producing very high resolution. So here we can see we are having T, S2, S1 and L. T belongs to the trailing electrolyte, and S1 and S2 are the two different samples, and L is your leading electrolyte. So when we apply the electric field into that, the electro migrate through the microchannel according to the electrophoretic mobilities. So initially your sample 1 and sample 2 will be in center. On the left hand side will be your trailing electrolyte, on the right hand side will be a leading electrolyte. But as the time passes, so in the center your sample 1 and sample 2 will be separated from each other, and your trailing and leading will be also on the different side of the electrolytes. So once a faster moving component separates completely from a slower moving one, it creates a regional of depleted charge between the two that increases the resistance and therefore local voltage in that region. So this increased voltage causes the slower component to migrate faster and close the gaps further. So isotopes could be used for separation of proteins as well as inorganic substances. Then further comes your immunoelectrophoresis, which is also used, very highly used with respect to antibodies. So antibodies are produced by immune system in response to foreign macromolecules, and each antibody binds specifically to one feature on one macromolecule, and this allows the use of antibodies for detection and quantitation of specific proteins in a complex mixture. So when electrical potential is applied to study of antigen antibody reactions, it is called immunoelectrophoresis. The antibody are electrophoretically separated and antigens diffuse towards each other, resulting in precipitin arcs where antigen antibody complexes form. And this technique has been referred to as immunoelectrophoresis. So antibody is placed in tough cut parallel to the direction of electrophoresis. So run the electrophoresis. As a result of precipitin arcs will be formed due to the antibody antigen complex formation. So in this method we do it in the 2% agar gel. An antigen mixture is applied into a small circular wells cut out of the agar. Initial electrophoretic separation is carried out depending on the charge and molecular weight. Then comes this one, we have discussed in detail already, isoelectric focusing. If you want to prepare from the examination perspective, you can go through these points one by one. So all the procedure is determined by the first we separate our proteins according to the pH. They find their isoelectric pH and then we migrate over the SDS gel. Yeah, so the same thing has been explained. Then this is your 2D gel, right? So that's it. This is one cartoon here with a paramecium parlor that we have discussed. You remember the restriction enzymes? So there's a fancy party is going on. They're saying that was the last year DNA invited the restriction enzymes to their Halloween party. So hi guys, great costume party. So he came with the mask over it with a drill within his hand, and he's about to cut these sweet DNAs there. Edu Fabrica. Day 6th. So yeah, now students, I will play one video, right? It will be about the whole thing that we have discussed. How can we use this from the practical perspective, how they are being used in the industrial. So it will be around 20 minutes video, so relax and enjoy that, and based on that then we will end our chapter of today of this Sunday, exciting Sunday over this point. Then I will start that thing once the things get uploaded, because it will interfere with the, or I will share this later. I play the video first. Bioprocessing, the purification. It's around 19 minutes video. Yeah, so let's start.
Today, we rely heavily on biotechnology for many of the necessities of life. For the food we eat, the clothes we wear, fuels for transportation, earth-friendly plastics, pharmaceuticals to treat our illnesses, and dietary supplements to help us stay healthy. Many of these products are produced using three core biotechnology processes: fermentation, recovery, and purification. Fermentation is basically cell farming. We program cells to produce a product. We nurture them as they grow and reproduce, and then we harvest them. In recovery, we separate our product from the cells where they were housed. And then in purification, we go a step further by removing everything else that's contaminating our product, leaving us with a very pure, concentrated solution. In this program, we're going to look at a typical purification process used in the manufacture of GFP, green fluorescent protein. GFP is broadly used as a biological marker because it's very well tolerated by most cells and doesn't interfere with normal cellular function. We'll examine the technologies, equipment, and materials used, how to prepare for the process, and how the GFP purification process is managed, step by step. There are two main operations used within purification: chromatography and filtration. Before we can appreciate exactly what these process steps are accomplishing, we need to take a closer look at what our green fluorescent protein has been through so far. Back in recovery, our harvested cells from fermentation were homogenized. Our target protein was inside each cell, so to get at it, the cells had to be ruptured. Homogenization freed a flood of cellular components, including membrane debris, cytoplasm, DNA, and proteins, including our target protein, GFP, and they were all mixed into the buffering solution used to suspend the cells. Most of these solids were then removed through centrifugation, or by centrifuge, but the liquid that remained, the clarified lysate, was still rich in biological products, including dissolved chemicals, proteins, and other impurities. Amazingly, through purification, we can target a single protein within this biological soup. In our GFP purification process, we'll be using multiple types of column chromatography. The column is a cylinder filled with glass, ceramic, or polymeric beads, which are engineered to interact with or bind with molecules based on one or more physical properties. Chromatography relies on differences. Each molecule has a unique set of physical characteristics, such as size, charge, or extent of interaction with water. Chromatography uses these differences to separate the target protein from other proteins and chemicals. Sometimes size is used to differentiate. Some beads have small holes in them and can temporarily trap or at least slow down smaller molecules as they travel through the column of resin beads, while molecules too large to enter the pores move around the beads and exit the column first. This type of chromatography is called size exclusion. In the case of charge, opposites attract, so a negatively charged chromatography bead will attract and bind to positively charged components in the process stream. Likewise, a positively charged bead will bind negatively charged components in the process stream. This charge-based chromatography is called ion exchange chromatography. And then there's water. Molecules that readily interact with and dissolve in water are called hydrophilic, water-loving, while those that don't are called hydrophobic, water-hating. Proteins contain regions that are hydrophobic and regions that are hydrophilic. Because water tends to form a shield around the hydrophobic patches within the proteins, they are not exposed to interact with the resin beads.
By adding salt to the protein solution, we remove the water shield, exposing these hydrophobic patches on the protein and resin so they can interact. This is how HIC, or hydrophobic interaction chromatography, works. In our process, which uses ion exchange and hydrophobic interaction chromatography, the chromatography equipment is housed on a skid to make it compact and mobile. The main part of the apparatus is a glass column filled with resin beads, but we also have pumps to move the clarified lysate through the process, a supply hose and port to feed the column, a pre-filter to remove any remaining particulates, usually solid cell debris that has not previously been removed, before the clarified lysate enters the column, an exit port for the processed solution, and auto switching valves for directing processed solution to either waste or collection. To help monitor the chromatography equipment and the solutions flowing through the unit during the process, a number of sensors are located along the product flow path. There is an electrical conductivity sensor at the column inlet, a pressure sensor just before the pre-filter to help identify a filter clog, a flow meter to measure the rate of solution movement through the column, and an air sensor to ensure that no air has entered the flow path. As a solution leaves the column, it passes a UV sensor that reads optical density, a second conductivity sensor, and a pH sensor that measures how acidic or basic the solution is. The conductivity sensors let us know when a new buffered solution has filled the column. When the conductivity reading on the exit of the column matches the reading from the sensor at the inlet of the column, then we know the new solution has completely displaced the old one. The ultraviolet UV sensor monitors the concentration of protein in the product by observing the optical density of the passing solution. This sensor works hand in hand with the valves on the exit of the column. Through the controller program, we can set a protein concentration threshold. When the optical density of the solution leaving the column is below the threshold, the valve directs the flow to waste. When the optical density of the solution leaving the column is at or above the threshold, this means that solution contains our purified product and the solution is directed to a collection vessel. TFF, tangential flow filtration, is at its heart a simple process step. We're going to pump a fluid through and across a special type of filter known as an ultrafiltration membrane. The size of the pores in the filter material determines what passes through and what's held back. As with almost any filtering process, we can choose what we want to keep. The solution that passes through the membrane is referred to as the permeate. Because the pores of the ultrafiltration membrane are small enough to keep the product from passing through, the permeate contains no product and is sent to waste. The portion of the feed stream that does not permeate the membrane is called the retentate. It contains the retained product and is the stream we are most interested in. What makes TFF different is a core technology that enables it to be faster, more efficient, more flexible, and even self-cleaning. In conventional or terminal filtration, a fluid is pumped directly into a filter. The particles within the stream that can't fit through the pores of the filter build up at the filter surface, eventually clogging it. In tangential flow filtration, the stream moves across the filter, that is, tangential to the filter, instead of directly at it. The cross-flow current actually picks material back out of the filter media or membrane and into the stream. This retained material, called retentate, is recirculated to the supply tank and will continue to loop through the filter for as long as the process runs. We'll be using TFF for two different tasks within the purification process: concentration and diafiltration. We are processing purified GFP from a chromatography step. The fluid from the chromatography step is purified green fluorescent protein dissolved in a buffering solution. In diafiltration, we add new buffer to the GFP retentate while displacing the old buffer, effectively exchanging buffer solutions. Note, the GFP is retained by the membrane. If we don't add a new buffer, then we're concentrating our solution. Concentration is simply the removal of water and buffer components from the feed solution that results in a more concentrated solution of green fluorescent protein. From operation to operation, or product to product, the purification process can look quite different. It could be as simple as a single chromatography step, one cycle of TFF to concentrate the product, and final filtration. Or it could involve several different types of chromatography, diafiltration between chromatography steps, and a final conventional filtration. Our green fluorescent protein process is pretty typical of the process for biopharmaceutical products. The clarified lysate will be pre-filtered, go through anion exchange chromatography, and the GFP collected and pooled. Next, ammonium sulfate will be added to make the solution high salt, followed by hydrophobic interaction chromatography, HIC. The GFP is then collected and pooled again. The solution then goes through a TFF ultrafiltration step, which includes concentration of the GFP followed by diafiltration to exchange buffer solutions and remove salt. And the process finishes up with final filtration into bulk bottles or bags. It's time to gather everything we'll need for the purification process and ensure that the area and equipment are ready to go. The chromatography skid and TFF system are checked for proper operation. Process hoses are attached and examined for leaks. We verify that we are using the correct column resin and that the resin is properly packed. The column is filled with a storage buffer, and the product path is checked for trapped air and purged if necessary. Our raw materials include the clarified lysate from the recovery process, various buffer solutions that are tailored to specific process steps, and ammonium sulfate that we add to one of the buffers to make it high salt. The purification process is managed through the use of a batch record. There is a separate batch record for each processing operation. These documents lead the operator through the process step by step, with each step requiring a sign-off and separate verification by a second operator. The batch record also includes spaces for documenting times, activities, operation steps, and instrument readings. Before the process can begin, the purification area must be cleaned, disinfected, and organized. Any unnecessary equipment or materials are removed. All equipment must be cleaned, sanitized, and set up as required by standard operating procedures. All required materials and documentation must be gathered and prepared before the process may begin. The purification process begins as the transfer tank of clarified lysate from the recovery process is connected to the inlet pump on the chromatography skid. The first chromatography step in our green fluorescent protein purification process is anion exchange. At this point in the process, the pH of the clarified lysate is about 8.0, which means that the protein is negatively charged. Because it is negatively charged, GFP will bind to the positively charged anion exchange resin. The pump draws the lysate from the vessel, past the first conductivity sensor and pressure sensor, and through the 0.45 micron pre-filter. The pre-filter removes any residual cell debris or other particulates that may have contaminated the solution. If the pre-filter begins to clog, the pressure sensor at the inlet side of the filter will register a rising pressure, and the controller will signal the need for a filter change. After pre-filtering and before the column, the lysate passes through a flow meter and an air sensor. Then, as the lysate passes over the resin beads, the negatively charged protein binds to the positively charged beads. The solution leaving the column passes a UV optical density sensor, a conductivity sensor, and a pH sensor. The optical density sensor's low readings confirm that the GFP is not in the solution, so the outlet valve sends the solution to waste. When all the lysate has entered the column, or when the capacity of the beads to bind the protein has been reached, it's time for elution. Elution is the release of, in this case, green fluorescent protein from the beads by using a new solution, in this case a buffer that includes NaCl, sodium chloride solution. As the new buffer is pumped through the beads, at some point the GFP no longer binds to the beads and is released into the buffer. The resulting product stream is usually referred to as the eluate. The UV optical density sensor, which measures protein concentration, indicates when product begins eluting from the column. At this point, outlet valves are switched to allow flow of the eluate, the product stream, to a collection vessel. When the UV sensor indicates that all of the GFP has come off of the chromatography resin, the outlet valves are switched to waste. When all of the eluate has been collected and pooled, the anion exchange chromatography step is finished, and it's time for HIC, Hydrophobic Interaction Chromatography. Hydrophobic interaction chromatography is based on the principle that hydrophobic chemicals on the resin surface will bind to hydrophobic patches on the GFP protein. In order for this to occur, the resin and protein eluate have to be in a high salt environment to remove the water shielding. The salt we use is ammonium sulfate. To remove the attached GFP protein from the HIC column, we simply lower the salt concentration during elution, causing the water shielding to reform, and the GFP protein detaches from the resin into the elution stream. The protein-rich eluate is collected and pooled. The product is now ready for the last major step, TFF. At this point in the process, tangential flow filtration will be used to concentrate and diafilter the GFP product stream. The eluate is rich in green fluorescent protein, but it's still too dilute and too high in salt. As the solution moves through the TFF apparatus, it leaves the supply tank, is pulled through a pump, past a pressure sensor, and then across the filter membrane. Everything that passes through the membrane, including the buffer solution, is known as permeate and, for this process, is sent to waste. The GFP protein is larger than the pores of the membrane and therefore is retained. The retained material, called retentate, is recirculated to the supply tank. Recirculation of the feed continues until the desired concentration of GFP is achieved. Following concentration, and while the protein solution is recirculating, a new solution, a storage buffer, is added to the feed. In effect, the protein is being washed by the flow of a new buffer solution in and the old buffer solution out. As this diafiltration step proceeds, the buffer solution that is being added to the feed replaces the buffer solution that the GFP was originally in, effectively removing any remaining salt as well. When this process is complete, the GFP solution is routed through a 0.22 micron final filter and then collected in appropriate containers, usually bottles or bags. The purification process is complete. The green fluorescent protein concentrate can now move downstream to final fill finish to be freeze-dried and packaged.
So that's it, students, for today. Everything has been shared over this, and I will share the later part of the lectures also with you. I hope you had a good learning of today, this protein, everything all around protein. Hope you could use these techniques in future somewhere. and remember me also. I will be very happy because it will be very nice, you know. You are all are graduate, postgraduate, PhD, somewhere you will be working. If you are getting something good out of these techniques after learning them, and you can bring out some change to the world, I will be the most happiest person that I could give a little iota of these knowledge, very little knowledge that I had with me at the moment. But if you can make it big to the world, I will be very happy. Yeah. So on that note, wish you all good night, sleep well. Tomorrow we will see us again. Our 4 days lectures are left now. That is, we will be finished by Tuesday then actually. So I do Fabrica. So yeah, 6 is over. So we have tomorrow chip and yeah, western blotting, southern blotting, northern blotting, and then we will do the facts analysis tomorrow. Yeah, two things will be covered tomorrow. That is your facts analysis and your southern and western blotting and your chip analysis to understand between your DNA and protein interaction. And the day 9th, we will deal with the bioinformatics part, X-ray crystallography, and a day 10th we will do the applications of these techniques that we have done. So by Tuesday we are almost over. Thank you very much for cooperation. If anyone missed their lectures, if anything have done anything, missed that stuff, you can come to these lectures again and watch them again. So I'm pretty sure that I have provided you all, and we are both learning from each other. It's not just I'm providing, I'm also learning from you all students. Okay then, good night, take care. Khub walo sir. Khub walo sir. Maybe it's in Bengali, I guess. I'm not sure. Um, Khub bhalo. I know this. Khub bhalo. I used to have, when I was in Gizan, a lady from West Bengal. Her husband was a professor in the Gizan University, and she really treat us like their son actually, the son and daughter. So yeah, yeah, yeah. So we used to go to her place, and she took us to the Durga Puja also during that time. I remember we make this sound something. So we also did with her on that day, and it was very. Yeah, I still remember those days. Very warm, very cheerful, and everyone was so happy and speaking a lot of food and lots of energy. I could see positive energy everywhere. Okay, then cool Balo show. And thank you so much. Tata, good night. Take care. Bye bye.